As you all know, the quest for a cure of HIV has begun. To date, there is one individual, Timothy Ray Brown, the Berlin patient, who has been cured through heroic actions, but impractical for achieving cure in only the general population of HIV-infected individuals. The cure field started with a lot of enthusiasm. The enthusiasm is still there, but the reality is that progress will be slow and will grind out over years to decades until we have a functional cure for a significant fraction of HIV-infected individuals. There are many pieces of the puzzle that need to be put together to solve the problem. One of them is, where does HIV persist and how does it persist despite very effective therapy? And we heard an important presentation from Francesco Simonetti on the persistence of HIV. We also need to know, is there a marker that we can use to indicate we're making progress towards a cure? So what was critical in the acceleration of antiretroviral therapy to its current state is a marker that substituted for clinical trials that relied on development of AIDS or death, and that was viral load, plasma HIV RNA. We do not have such a marker for cure. We don't know whether we're moving successfully down the field towards a cure or not, and we don't have a practical means of measuring that. What we're resorting to is a treatment interruption or a intensely monitored antiretroviral pause, or IMAP, in which we look for initial evidence of rebound. And we have two very important presentations, one from Jonathan Lee and the other from John Frater, about initial analyses of potential biomarkers that would predict the time to rebound after stopping ART, with the goal being that if we can identify a biomarker, affect that biomarker, i.e. lower it, that we could understand whether we're making progress, in that if we lower the biomarker, and that biomarker is associated with a delayed time to rebound, then we would know we were making progress. I hope that's clear. And lastly, we're all thirsty and hungry for interventions now that might get us closer to a cure. And in this field, unlike the field of antiretroviral therapy development, in this field, the primate model, the non-human primate model, seems to be a very important tool to test out new strategies that are really impractical to test out in clinical trials. Primate models can be set up, and experiments can be done in 6 months to a year. The average clinical trial takes 2 to 3 times that, and is resource intense and is associated with risks. So we're going to hear at the end from James Whitney, who's going to describe an agonist of a toll-like receptor 7 as a potential intervention that can reduce viral reservoirs in the primate model. So I'm going to start by asking each of the presenters, first Francesco, then John Lee, then John Frater, and then finally James Whitney, just to give a 2-minute encapsulation of the key take-home messages from your presentations, and then open it up to questions.
Okay, so good afternoon, everyone. So in the last couple of years, we observed, us and other independent groups, that infected cells expand and survive over years under therapy, but there was an unanswered issue whether these expanded clones are a true mechanism of persistence. There's a recent paper on Cell. They looked for a lot of clones. They couldn't find any infectious provirus. So we observed for a long period a patient that had a residual viremia made of just one viral clone. So we observed a match between DNA and RNA of this clone, and so we thoroughly characterized this provirus, and the full genome, it looks completely non-defective. And we perform a couple of experiments. An infectious virus recovery experiment, taking the cells from the patient. And also, we transfected cells with the sequence of this provirus, and we proved that it's infectious. So, leading to the answer that these expanding clones can harbor non-defective proviruses that can make infectious virus. Moreover, this patient also had a diagnosis of a squamous cell carcinoma of his tongue, and there was a really sharp temporary link between the tumor progression and the rebounding viremia. And we observed an enrichment of this particular clone in the tumor tissue cells, suggesting that antigenic stimulus might be involved in this expansion.
Thanks. So just some key points here, that we've learned that an HIV-infected cell can persist on antiretroviral therapy. We knew that for a long time. What's new is that infected cell can not only persist, but it can multiply and expand. And Francesco's work shows that that expanded cell clone carries an infectious virus that can fuel rebound when we stop therapy. So we're not just dealing with cells that were infected and remain quiescent for a long period of time. They can actually proliferate, expand, and are a bigger obstacle to cure than we initially thought. And we're going to have to, as Dr. Hughes in the audience said in his plenary, we're not only going to have to block all viral replication, we're going to have to block the replication of these infected cells and kill them. Okay? So why don't we move on to John Lee?
So the discovery of biomarkers has really been instrumental to improving the lives of HIV patients. And as John mentioned, the discovery that viral load as a biomarker for risk of disease progression led to its use in clinical trials as a surrogate endpoint, leading to shorter, smaller studies, greatly accelerating the pace of drug development. As the HIV field is increasingly turning towards searching for strategies for to delay HIV rebound for this sustained ART-free HIV remission, we now need biomarkers that can help accelerate those studies as well. We performed a pooled analysis of a number of AIDS clinical trials group analytic treatment interruption studies, or ATI studies, and we evaluated both the HIV rebound kinetics after patients stopped therapy, as well as looked to see whether the levels of various HIV reservoir measures could predict the timing of viral rebound. And what we found first was that the viral rebound kinetics after treatment interruption was that most patients did rebound by week 4, but that there was a subpopulation of patients with more delayed rebound, that patients who were treated during acute and early infection rebounded quicker, that those who were treated, or sorry, rebounded later, and that those that were treated with an NNRTI class of medications also tended to have delayed rebound as well. When we evaluated the HIV reservoir, we looked both at the HIV DNA level as well as this active reservoir, the production of HIV RNA, as well as the production of plasma HIV RNA and cellular HIV RNA. And what we found was that this active HIV reservoir at the time of treatment interruption could help predict when patients rebounded, had viral rebound during the treatment interruption, and that those individuals with delayed rebound had significantly lower levels of cell-associated RNA and residual viremia by a single copy assay. And what this really implies is that the quantification of this active HIV reservoir could provide biomarkers of efficacy for therapies that aim to achieve this ART-free long-term HIV remission.
So what's really exciting about this is John has preliminary evidence that simple measures, the number of copies of RNA in cells and the number of copies of RNA in plasma, this sounds very familiar, right, could be a marker of success and progress towards a cure. If we lower both of those, we might get closer to a cure. In other words, the duration after stopping ART before virus comes back would be longer. And this is incredibly important and very analogous to the situation where when we were treating people initially with antiretroviral therapy and dropped viral load, that was associated with lower likelihood of AIDS and death. So it's really quite an exciting finding. It's just a beginning, though, as John said in his presentation, and we encourage you to write about this as a critical factor in progress towards a cure. Many of the funding agencies were skeptical that we would ever be able to find a biomarker, of a simple biomarker of progress towards a cure. And with that, I'm going to turn it over to John Frater, who did some similar analyses, but in a different group of patients.
Okay, so thank you very much, everybody, and good afternoon. So again, our work is focused on this concept of what happens when you stop antiretroviral therapy and when does your virus come back, and particularly inspired by the Visconti cohort that you may be familiar with. In patients who are treated in early infection, there seems to be something special about patients early on. If you treat early and stop therapy, it is completely different to what happens when you treat a chronically infected patient and stop. And in Visconti, we know there are about 14 patients who have not rebounded many years after stopping therapy. Our work comes from a study called SPARTAC, which was a large trial of antiretroviral therapy in primary infection. 360 patients in all recruited over a number of continents. And our work is a sub-analysis of patients in that study who had treatment for 48 weeks in primary infection. Within about 10 weeks of being infected, they went onto ART for 48 weeks, and then they stopped. This time last year, we presented data showing that if we measured DNA levels, HIV DNA levels, at the moment that you stop therapy, that predicted time to rebound. And we know that in these patients, 14% were undetectable a year after stopping. So there is something different about these patients. They're not behaving like you'd expect them to. So the study we've just presented is taking this work a bit further and looking to see are there other markers as well as DNA that might help us predict what happens when you stop therapy. So very similar to the work that Jonathan has been doing. We've focused more on the immunology. We've been interested in HIV immune responses and then markers like activation markers, CD38, HLA-DR, and then more specifically markers called immune checkpoint markers or exhaustion markers that have lit up the world of cancer medicine and seem to be really important there. Markers PD-1, LAG-3, and TIM-3. And when we measured these markers in our patients, what was interesting, at the moment that they stopped therapy, 48 weeks after treatment, and they stopped therapy, the markers didn't really tell you very much. But if you then went back and actually measured those markers from samples on the day they started therapy, the three markers that stood out were PD-1, LAG-3, and TIM-3, these exhaustion markers, these immune checkpoint markers. Activation and the other markers that we thought might matter didn't matter, but these three things stood out. It's hard to explain why, but it opens up a number of very interesting questions and hypotheses as to what is the nature of these cells, that when you start therapy, there is something inherent about them, that when you stop therapy, these cells wake up and other cells don't. So I think this adds to the data, that DNA helps predict rebound. These three markers, again, open questions as to mechanism, which we really don't understand. Why does the virus not come back? Understanding these markers will help that. But also, it'll help build an algorithm to help try and predict patients who've been on therapy from primary infection maybe for 2 or 3 years. If we measure these sort of markers, we're able to say, well, you can come off and see how you get on. You may do well. We don't know the answer to that yet, but it's the first steps.
Thanks, John. So these markers that John was talking about, this is during the acute untreated infection. And if the immune system shows signs of exhaustion, okay, as manifest by expression of PD-1, LAG-3, TIM-3, if it shows signs of exhaustion, that remarkably, after suppression with antiretroviral therapy, that the degree of exhaustion of the immune system predicts how quickly the virus comes back. And I would interpret it as in those individuals whose immune system has said, you know, we can't handle this infection. We're shutting down. We're shutting down the immune response. Those are the ones who are going to rebound quickly. And just to put it in the right context, cancer therapy has been revolutionized by blocking these receptors. So some individuals with cancer have high levels of exhaustion markers. The tumor progresses, but by reversing that exhaustion, waking up the immune system, empowering it to respond, has resulted in very dramatic responses in cancer. And it also suggests, although John didn't imply this because he's very careful not to go beyond his data, it suggested blocking these receptors in HIV infection might improve outcome. And there is a clinical trial going on to look at blockade of one of the receptors, PD-1 and PD-L1. So that's very exciting new information. So I'm going to turn finally, before opening up to questions, to Dr. Whitney, who presented exciting animal model data on a new, potentially, a new intervention that might get us closer to a cure.
Thanks, John. So I guess one of the major motivations in the HIV cure research field, obviously other than eliminating HIV in infected patients, is looking at different modalities to try and reduce the burden of virus in patients. So one major research thrust, as I mentioned in my talk, is to try and juxtapose shock and kill or kick and kill therapies to try and reduce the viral burden. So a lot of the work that has been done historically with virus activators, they've basically repurposed a lot of oncology agents, typically HDAC inhibitors. And while these are potentially of some utility, they do carry a lot of baggage for reactivating virus in the fact that they are somewhat toxic and quite immunosuppressive. So the preclinical study that we undertook in association with Gilead, we didn't really want to go that particular route. So we opted to use something a little more direct, TLR agonist, and in particular TLR7 agonist. Now TLR7 impinges directly on viral RNA, so it's kind of a virus-specific type of modality. And what we found, we started a 10-monkey study. The animals were infected with SIV, and we put them on a very, very suppressive regimen, also with the help of Gilead. And that regimen was critical to align our monkeys and make them very, very comparable to the types of suppression you see in clinically treated HIV-infected patients. Once those animals were suppressed, we dosed them with TLR7 agonists. And what we found was that this particular modality has a number of positive features. We get some kick, but we also get some kill in that TLRs also, first of all, they potentiate immune responses. So we enhanced overall immunity in these monkeys. And then the really novel conceptual finding was that after repeated dosing, the animals actually, upon dosing, elicit viremic blips. And that tells us that basically we're unleashing or unrestraining the latent reservoir. And the magnitude and size of these blips is much greater than we've seen with other agents, such as HDAC inhibitors. So really the important finding for us is that we have something that is really non-toxic, it's immune potentiating, and it also elicits very high level of viremic bursts. And so the outcomes that we measured, obviously we had reduced amounts of reservoir in these animals in a number of tissue compartments. And then when we stopped ARV therapy, the treated animals had a new and reduced set point in comparison to control animals. So that's essentially it.
So again, this compound that activates a receptor, TLR7, it's being studied in hepatitis B to try to cure hepatitis B. There is proof of concept in a few woodchucks who are a good model of hepatitis B infection, and also a response in chimpanzees. There's a phase 2B trial of this compound, GS9620, in hepatitis B. And based on James's work, there is a clinical trial of 9620 starting this week at 3 clinical centers in the United States. Okay? What it appears to do is 2 things. It wakes up latent virus, causing virus production to the point where it's easily detectable in plasma. It seems to decrease the number of HIV-infected cells in tissues, okay, which is a very favorable thing if that can be confirmed by additional studies. And when therapy is stopped, it appears to delay, it appears to result in less virus coming back, less viral replication. So 2 things or 3 things really in the right direction of where we want to go. And it's very exciting because we are going to try to translate this preclinical work in primates into human beings very shortly.